First of all, the dendritic cells, DC, play the role of sentinels in the mucosa. They are part of the antigen-presenting cells. As soon as they encounter antigens from pathogens, they become activated and migrate to the secondary lymphoid organs, where they present the antigen to lymphocytes, which in turn become activated. Dendritic cells represent the link between the innate and adaptive immune response.
T lymphocytes belong to the adaptive or specific immune system. Thanks to their surface receptors, TCR, they are able to recognize specific antigens presented by antigen-presenting cells.
Several subsets of T cells exist. T helper cells, or CD4 plus T cells, allowing the differentiation of B cells into plasma cells or memory B cells, or the activation of T cytotoxic cells and macrophages. T cytotoxic cells, or CD8 plus cells, allowing the destruction of virus-infected cells and tumours. T regulatory cells, formerly known as suppressor T cells. They are the engine brake of the immune system, crucial for immunological tolerance. And finally, memory T cells persist for a long time after an infection. B lymphocytes also belong to the specific immune response. They express surface receptors, BCR, able to recognize a specific antigen. B cells can differentiate either into plasma cells, able to secrete antibodies, or into circulating memory B cells.
Antibodies play an important role in the humoral immunity. They can flag an antigen at the surface of a pathogen, a process called opsonization, facilitating its phagocytosis by the granulocytes. Several subsets of antibodies exist. IgA are the most important immunoglobulins for mucosal protection. They are found in the serum and in secretions, tears, saliva, colostrum, and mucus. IgG are important for pathogens opsonization. They represent 75% of immunoglobulins of the serum. IgE are involved in allergic reactions. They are also involved in fighting parasitic helminth infections. IgM are the first immunoglobulins produced by the fetus and virgin B cells. Thanks to their pentameric structure, they are very efficient in triggering the lysis of microorganisms. IgD are primarily found at the surface of B cells and allow the binding of the antigen. Finally, natural killer cells belong to the innate immune system. They provide a fast response against virally infected cells and tumor cells. They are able to kill those cells directly by inducing apoptosis or by secretion of cytokines that may eradicate the infected cells.